Thank you so much for the kind introduction, and I would like to thank the organizers and Bart Lambrecht especially for the kind invitation to be here in the face and in the era of COVID-19. And it's a great pleasure being in a cross-disciplinary conference dealing with translation and immunology. And I would like to share with you, acknowledging that I'm the last talk in a very long day with many exciting talks, I would like to share with you some of our excitement concerning the current state of play in the complement drug space and specifically the therapeutic complement inhibition. And I would like to share with you and walk you through the latest clinical developments, the key milestones that have led to our reshaped understanding of the role of complement in human pathophysiology. And on the way, I will hopefully dissolve some of the long-held misconceptions and myths surrounding complement C3 inhibition and provide some novel insight in the ongoing clinical efforts. Before moving into the clinical front, I would like to pay my personal tribute to evolution, pay my tribute to the evolutionary perseverance of the complement system as one of those innate immune systems that transcend the evolutionary ladder from lower vertebrates up to mammals, higher vertebrates, and humans. Complement actually activities have predated higher vertebrates by more than 900 million years, so that's a lot of evolutionary time. And activities have been characterized all the way down to sponges and sea urchins. And what I would like to pay your attention to is this particular species of fish, the teleost fishes, where the Lambrecht group and other groups have done seminal studies showing that a lot of complement proteins appear and emerge in multiple isoforms in this species, generated what is known as immune diversity in the face of an underdeveloped adaptive immune repertoire which exists in these organisms. So in compensating for a very low humoral response and a very underdeveloped adaptive immune response, these species have a very well developed and diversified complement system. So I would like to summarize in this slide the current perception of the complement system as a key effector of innate immunity and inflammation. It's quite a complex and dynamic network consisting of a lot of glycoproteins interacting with each other in a cascade-like manner. But I would like to focus your attention on the fact that, and remind you of the main canonical routes of triggering, which are the classical and lectin pathway triggered by specific carbohydrate and antigen antibody complexes targeting surfaces. And of course, I would like to mention the evolutionary, more ancient alternative pathway, which is the one responsible for the low rate of activation of C3 in the circulation through the spontaneous so-called takeover mechanism by which C3 gets hydrolyzed at a very low rate. And this is what keeps the pathway in a standby mode ready to be triggered and triggered into action whenever a pathogenic invasion happens. So the alternative pathway, which actually covers most of the effector functions by being the mechanism that amplifies the complement response, all the routes of activation converge at the proteolytic cleavage of the central protein called C3, which is a highly abundant protein in serum, and this leads to the proteolytic cleavage of C3 and the release of a lot of bioactive fragments that opsonize the target surfaces and also trigger a lot of immunomodulatory functions, including phagocytosis, adaptive immune stimulation, a lot of chemotaxis being triggered by complement byproducts, and all this ends up in recruiting a lot of immune effector cells close to the bacterial challenge in order to contain the microbial intruder. And this is the traditional perception of the complement system as we know it from the textbooks of immunology. And the end product of this route of activation is the formation of this multiproteinaceous lytic pore, which is called membrane attack complex, or MAC, or C5p9, which actually osmotically lyses the target cell. So this is the traditional aspect, but I'm summarizing a lot of studies in this slide showing you what is our current perception of the complement system. We like to believe that the complement system is actually a very fine modulator or orchestrator of a lot of functions and biological processes that happen at the interface of innate and adaptive immunity. Complement proteins and pathways are known to interact with a lot of other biological pathways, including the coagulation system. There are reciprocal interactions activating one or the other. A lot of interactions with the TLRs, with different compartments in the bone marrow. A lot of insight has been produced in showing that complement plays a role in tissue remodeling, tissue repair, and angiogenesis. And of course, there is not a single cell type that does not interact with complement receptors or complement activation fragments as shown on this slide. And this is actually, this actually exemplifies our current perception of a systems-wide notion of the complement system in participating in many, many different functions that have to do with innate or adaptive immune responses. But before moving on to the clinical front, I would like to spend the next couple of minutes showing you a few of the so-called paradigm shifts that have reshaped our understanding of complement biology. And by saying that, I would like to stress the fact that these paradigm shifts, in essence, have actually reshaped the field of complement therapeutics by opening up new prospects for clinical application in different clinical conditions. And I would start with these slides showing that the seminal work by the late Panagiotis Tsonis in collaboration with John Lambris, where they showed that C3, the complement proteins C3 and C5 are specifically expressed in the regenerating zones of this spectacular animals called urodil amphibians, which actually faithfully regenerate every single part of their body, including the lens and the limbs. C3 and C5 were specifically found to be expressed in the regenerating zones of this lower vertebrates, and this information was actually carried over to the mammals, where we were able to show with John Lambris quite a few years ago, and with other collaborators, that complement activation products play a key role in triggering liver regeneration under specific settings of acute hepatotoxicity or liver regeneration or partial hepatectomy. So what our current understanding of the complement system in the liver space is that it actually triggers a regenerative response which focuses more on the Kupffer cells, the liver macrophages, which express complement and aflatoxin receptors, and this function actually leads up to the release of key cytokines such as IL-6 and TNF-alpha, which in turn trigger a regenerative program in otherwise quiescent hepatocytes through the activation of the STAT3 transcription factor, leading up to hyperplastic proliferation of the remnant liver and triggering all this process which is known as liver regeneration. Another paradigm shift that has actually created a lot of turmoil and has sparked a lot of interest in pharmaceutical development in developing complement therapeutics as therapeutic regimens in cancer immunotherapeutic protocols is the realization about 10 years ago that the complement system not only participates as a cytolytic augmenter of the cytolytic action of monoclonal antibodies in tumor immunotherapeutic protocols, but it can actually do the other way around. It can actually modulate anti-tumor immunity by promoting tumorigenesis through a diverse set of immunosuppressive mechanisms. And this slide, published in a review in Nature Reviews Immunology, summarizes the effort of different labs, showing very nicely and eloquently that complement activation fragments can actually modulate the immunosuppressive tumor microenvironment and create an environment that actually promotes tumor progression and also promotes the invasion of tumor cells into the circulation and their metastasis to distant organs. Complement was shown to be deposited in the tumor vasculature in different tumor models. Specific activation fragments were identified early on by the Lambris and Kukos groups back in 2008 in a seminal Nature Immunology paper as being responsible for the recruitment of myeloid-derived suppressor cells to the solid tumor and the suppression of T-cell anti-tumor T-cell responses. And of course, this led to the first proof of concept studies of therapeutic blockade of the C5a receptor in mice by the PO group. And very soon, this led up to the first clinical effort by Innate Pharma and AstraZeneca in putting together phase 1 and phase 2 clinical trials to test complement therapeutics as immunotherapeutic regimens in combinatorial immunotherapies for cancer. Of course, there is a lot of evidence suggesting that C3, upstream inhibition at the level of C3 may have broader therapeutic efficacy in different cancer models. A lot of work has been done by Alberto Mantovani, Cecilia Garlanda, in collaboration with John Lambris, in showing that C3 inhibition has therapeutic efficacy in skin epithelial carcinogenesis and specific sarcoma models. So I would like to also pay tribute to this paradigm shift that was set off by the elegant work done by Beth Stevens in collaboration with John back in 2007 and published in Cell at that time, showing that complement activation can actually modulate the synaptic circuitry of the developing brain by engaging microglial cells and astrocytes in what seems to be the so-called synaptic pruning of weak synapses in the developing CNS. And this function was had fundamental importance because later on a lot of pathophysiological models were developed where it was clearly shown that this microglial astrocyte complement axis gets aberrantly reactivated during disease in several neuroinflammatory or neurodegenerative contexts, leading up to the so-called increased synaptic loss that is accompanying cognitive decline in several neurodegenerative diseases. And this has been an active field of investigation for new complement therapeutics too. Also, Nature Reviews Immunology celebrated this finding a few months ago by releasing an editorial article in its special issue on the most important immunological breakthroughs in the last 20 years. So this work was actually highlighted there. And I will move on to show you what is the current state of play in the field of complement therapeutics and complement-associated diseases. The list of complement-associated diseases is long, and it's getting even more and it's getting even longer as we speak, because it's being realized that complement dysregulation or excessive activation can lead to detrimental tissue inflammatory effects that can cause tissue damage and chronic disease, chronic pathology in different diseases of the acute or the chronic spectrum, in hematological diseases such as PNH, in neuroinflammatory diseases such as neuromyelitis optica, in ocular inflammatory diseases such as AMD, and I will try to walk you through some of the latest clinical developments in this space. Of course, a lot of effort has been placed in characterizing complement as a potential therapeutic target in neuroinflammatory diseases, such as Alzheimer's and Parkinson, through this aberrant reactivation of the axis that leads to the synaptic loss of a lot of synapses from the central nervous system. And of course, complement has been recognized as a key perpetrator of the so-called autoimmune driven neuromuscular diseases such as myasthenia gravis and the disease called neuromyelitis optica. A lot of work has been placed in this field, exploiting new complement therapeutics in this direction. The complement space is being rapidly expanded with the approval of new complement drugs, and this is really important to stress because it has been a long time since the first approval of the so-called first complement-specific drug back in 2007, the monoclonal anti-C5 antibody Soliris. And 14 years later, we are witnessing a drastic reshapement of the landscape with the approval of the first C3 targeted therapeutic called Empaveli, and the recent addition to this complement arsenal of a new therapeutic, Avacopan, which is a small molecule C5a receptor specific antagonist, which has been recently approved by FDA for the rare renal disease ANCA-associated vasculitis. So we have a lot of potential therapeutics to be exploited in different diseases, and we're moving even closer to what is considered as tailored complement intervention. First of all, I would like to stress that the so-called blockbuster complement drug that was approved back in 2007, Soliris, was deeply rooted in academic effort that was led by John Lambris, Brigitta Stockinger, and Yvonne Fry back in the middle 80s in the Basel Institute of Immunology, when they were the first to isolate the first monoclonal antibodies against MAU-C5, and later on these antibodies were used by investigators affiliated with Alexion to show that there is therapeutic efficacy of blocking C5 in specific inflammatory disease models, and this led to the approval of Soliris. And of course, Soliris is a blockbuster drug, but also one of those rare disease drugs which are listed as exuberantly priced. It has a great socioeconomic burden especially in developing countries. And I would really encourage everyone to read this famous Bloomberg article suggesting that rare disease drugs are considered like a gold mine for pharmaceutical companies developing such complement-specific therapeutics. Of course, we have a multitude of complement-specific therapeutics being developed across the corporate R&D pipeline, targeting different components and different pathways. And of course, this explosion of therapeutics in the recent years, leading up to clinical studies in different phases of development, has been associated at times with what we call a quite obscure development of corporate drug development, which is not really associated with the release of the scientific literature in the public domain. So taking this opportunity, I would like to bring to your attention this recent commentary that we co-signed with a lot of complement investigators in Nature Immunology back in 2019, suggesting that we have to work a little bit more intensely in releasing the scientific evidence and the research results that are supporting corporate drug development. For the rest of my talk, I would like to focus on the story the long story of the so-called compstatin family of peptidic C3 targeted inhibitors. This new class of therapeutics has been brought to the spotlight lately because of the approval of Empaveli, which is a second-generation inhibitor of the same family. But the story started back in 1996 with the discovery of a peptide which specifically binds to C3, complements C3, and blocks its activation. And this peptide gave its name to an inhibitor family encompassing several peptides, cyclic peptides of 13 to 17 amino acids long. And these peptides act as protein-protein interaction inhibitors that actually block the access of C3 by binding to C3 and C3b, block the access of native C3 to the C3 convertase, and thereby inhibit C3 activation and opsonization of target surfaces. What I would like to stress is that these peptides have an exquisite species specificity, binding exclusively to human or non-human primate C3. And this has actually, in a way, accelerated their clinical development. And here I'm summarizing a long effort over the last 25 years, starting from 1996, when John Lambris and his collaborators discovered compstatin using phage display library, and leading up to the recent FDA approval of Empaveli, a second-generation compstatin-based therapeutic that was approved for treatment of PNH. And this long and rich history of development has encompassed a lot of effort by many, many collaborators who work towards the modification of the original peptide through structure-activity relationship studies. A lot of molecular and structural work has been placed in developing novel leads that were advanced to clinical trials and proof-of-concept studies in different indications. And I would like to give you a few examples concerning this third-generation compstatin inhibitor that is already in phase 2 development and clinically developed by Mendes Pharmaceuticals, the so-called AMY-101 C3-based therapeutic. This inhibitor has a very promising clinical profile. It has a very high affinity, binding affinity for C3, which is almost 6,000 times greater than the original compstatin that was discovered in 1996. It has a very good solubility profile and an exquisite plasma half-life, which is quite long for peptide therapeutics, which exceeds over 50 hours in plasma, and favorable toxicology and safety profiles in first-in-human studies. The first disease that I would like to give you as an example is PNH, which is the archetypal complement-mediated disease driven by the dysregulation occurring on the surface of a specific set of erythrocytes, a clone of erythrocytes that lack specific complement regulators called CD55 and CD59. These regulators lead to uncontrollable complement activation on the target of these cells, leading to spontaneous lysis, chronic anemia, and this has been the disease for which Soliris was first approved in 2007. However, the pathogenic mechanism that remained for many, many years unaddressed by anti-C5 therapy has been the so-called C3-mediated extravascular hemolysis. The fact that these cells get rapidly and continually opsonized by C3 fragments remained unaddressed by anti-C5 therapy and led to the extravascular clearance of these cells in patients that did not respond to Soliris. So in addressing this clinical need, AMY-101 was tested in vitro in models of PNH and showed efficacy in blocking both intravascular and extravascular hemolysis. Both of the processes were fully blocked by AMY-101 treatment, and this led to the recent clinical trials that were recently completed at the phase 3 stage and led to the FDA approval of the second-generation compstatin analog Empaveli or pegcetacoplan, which was nicely compared in a head-to-head comparison with Soliris in a phase 3 study that was recently published in New England Journal of Medicine. I would shift gears here and show you a little bit of what is going on in this space, the space concerning the development of complement therapeutics for dry AMD, this disease which remains without any approved therapy at hand currently. Dry AMD or geographic atrophy has been linked to complement alternative pathway dysregulation through genetic variants that have been associated with this dysregulation. It's a chronic disease of the retina responsible for irreversible vision loss, and it's a highly prevalent condition affecting more than 200 million individuals across the world. And recent studies have shown that a compstatin-based inhibitor of the family of compstatins, APL2 or pegcetacoplan, as it is known, or Empaveli, as its commercial name is now known after the approval by FDA. This inhibitor provided first efficacy data in 2 phase 3 studies. In one of them, it actually met the primary clinical endpoint of reducing geographic atrophy lesion in this patient, and in the other phase 3 study, it narrowly missed the point. And clearly, it has the potential to become the first treatment, anti-complement treatment for geographic atrophy. What I would like to show you is what we consider even more exciting is the fact that we have now, Dr. Lambris has been developing a 4th generation inhibitor of the compstatin family called AMI-106, which shows even more favorable characteristics for development as an anti-inflammatory treatment for ocular diseases. This compstatin analog has prolonged intraocular residence and has a very nice tissue penetration profile inside the retina. You can see here immunostaining showing very nice colocalization in the back of the eye, in the choroid or the RPE-Brooks membrane interface, where exactly this inhibitor binds locally expressed C3 or C3 coming in from the circulation. So these data are really giving us a lot of excitement in trying to see if we can have a best-in-class C3-based inhibitor in this highly prevalent disease. And I would shortly move to COVID-19 associated ARDS. Mika and Netea gave us a very nice overview of the pathophysiology of the disease. I would like just to stress that early on during the first wave of the pandemic, we formulated a hypothesis together with an international group of collaborators, suggesting that it would be really beneficial for these severe COVID-19 patients to target the activation of complement at the C3 level, because we thought that this would be upstream of the macrophage activation level, suggesting that this could really ameliorate the detrimental thromboinflammatory reactions that are mediated by the hyperactivation of innate immune pathways in these patients. And these hypothesis was quickly put to test by a small compassionate use program in Italy, in San Raffaele in Milano, where a case series of 3 patients were administered AMI-101 for 14 days and showed a rapid decrease in inflammatory markers in their circulations, which was accompanied by a rapid improvement of lung function and resolution of ARDS within 14 days after administration. And what was even more interesting was in a retrospective study performed between this small case series and a small patient cohort treated with eculizumab, Soliris, we were able to show that we have a distinct activity profile of this inhibitor, showing a broader effect, a broader inhibitory effect on a thromboinflammatory pathway, which has a key role in severe COVID-19, and that is netosis. And we were able to show that we can intercept platelet and neutrophil activation, leading to increased netosis in these patients and in COVID-19 immunothrombosis. And these results collectively led to the conduct of a first phase 2A, phase 2 randomized placebo-controlled trial in severe COVID-19 patients presenting with ARDS, for which the interim results were recently released. And I would like to share with you an initial set of results from the interim analysis of this phase 2 trial of AMI-101 in severe COVID-19, whereby a higher proportion of AMI-101 treated patients at this stage have met the primary clinical endpoint, adding a benefit of almost 25% on top of standard of care and dexamethasone. AMI-101, this C3 therapeutic, appears to have elicited sustained and complete C3 inhibition throughout the treatment window, and there appears to be a broad impact of C3 inhibition on thromboinflammatory pathways, with more prominent impact on mean netosis levels in these patients treated with the C3 therapeutic. I will slowly move on to the last example of a clinical development program, which is even more exciting to us and has been more fascinating in our eyes, because it really links to a disease which has been studied in terms of developing anti-inflammatory therapeutics, but it's a very prevalent disease that leads to the destruction of the tissues that support the teeth. The disease is called periodontitis, periodontal disease, and it's a highly prevalent chronic disease that afflicts about 50%, almost 50% of US adults, and its severe form is associated with an increased risk for certain systemic inflammatory comorbidities, including atherosclerosis, rheumatoid arthritis, Alzheimer's, and even diabetes. In this context, the Lambris and Hatchis and Gallis groups at the University of Pennsylvania were able to show over the last 10 years in preclinical proof-of-concept models that C3 activation plays a central role in the inflammatory response that perpetuates the tissue-destructive force instigated by the transformation of a symbiotic microbiota in the oral cavity into a dysbiotic microbiota that feeds on this inflammation and creates a vicious cycle that is perpetuated by complement C3 activation and leads to the osteoclastogenic alveolar bone loss that afflicts this patient. And in interesting proof of concept studies in non-human primate models of natural occurring or induced periodontitis, the Lambris and Hedges and Gallis groups were able to show that a local injection, intragingival injection of this C3 therapeutic, AMI-101, could reduce very important clinical indices of periodontal inflammation, including the gingival index and bleeding on probing, and also inhibiting the so-called osteoclastogenic activation in this condition that leads gradually to the alveolar, to the bone loss, which afflicts the patients. And this effect was really sustained and pronounced even 6 weeks after the discontinuation of treatment. These results were readily translated to the first human trial in patients with gingival inflammation, for which the results were recently released and endorsed by a number of almost more than 30 clinical experts in the field of dental medicine. And I would like to show you this recent story, this opinion article that came out in Trends in Immunology a few weeks ago, and actually made it to the cover. And this is really interesting to note that a mainstream immunological journal gave its cover to a story concerning periodontal disease. And that speaks to the promising potential of these therapeutics in this space. And here I'm just summarizing the results that were recently published in the Journal of Clinical Investigation from this first phase 2A study, placebo-controlled study of AMI-101 in patients with gingival inflammation. And I would like to point your attention to these graphs showing a significant drop in key clinical indices of gingival inflammation, including the gingival index in these patients, the so-called bleeding on probing, and of course a broader effect of C3 inhibition on the release of metalloproteinases such as MMP8 and MMP9, which have been implicated as significant prognostic factors for progression to the disease, to the severe disease form, which is periodontitis. So AMI-101 has the potential to become the first host modulatory therapy that exploits complement modulation for all inflammatory diseases. I believe I have shown you the potent and durable anti-inflammatory effects of AMI-101 in human patients with gingival inflammation, and these results collectively pave the way for the future phase 3 trials of AMI-101 in periodontal diseases and other diseases such as peri-implant diseases where complement has also been implicated. And finally, for the last couple of minutes, I would like to take your time in order to convince you that some of the myths and misconceptions in the field are really should not be there and should actually be placed in the right context. For many, many years, the field has been dominated by the notion that anti-C5 therapy is the only way to go around complement modulation at the therapeutic level. But C3 inhibition is quite feasible, and this has been proven by many clinical studies, and actually the recent FDA approval of Empaveli has solidified this profile by showing that we can actually saturate a highly abundant plasma protein such as C3 by injecting small-sized inhibitors in the circulation of patients. So technical feasibility aspects have been done away with, and of course the safety aspects regarding the safety of C3-based inhibition need to be placed in the right context. And by saying that, I would like to leave you with these take-home messages, really moderating the discussion around how risky is C3-targeted therapy. The clinical experience so far with all the C3-based inhibitors used in patients with PNH have shown that there is a very good safety profile. No more susceptibility, there's no susceptibility, greater susceptibility to infections compared to eculizumab. And of course, the FDA approval of Empaveli has solidified this safety record. Of course, for acute treatments, does not raise any concerns as far as C3 therapeutic inhibition is concerned. Most of the concerns and reservations have been originated from the so-called primary C3 deficiencies in patients, which show susceptibility to infections. But this has to be stressed that it is observed mostly in young patients, in children, and patients that have not reached adulthood. What we believe is happening here is a compensatory, dynamic response of the immune system. So when the human immune system develops into adulthood, the role of the complement as a pathogen immunosurveillance system probably subsides and gets compensated by other parallel systems. And of course, we have to make a note that pharmacological C3 inhibition in no sense can can phenocopy the genetic C3 deficiency and the compensatory mechanisms that develop there. Of course, there's a lot of discussion around intracellular C3 and the fact that it can actually provide a lot of sustaining survival signals for developing immune responses in the absence of plasma C3. Small amounts of uninhibited C3 can still support opsonization. And of course, there are several non-complement related mechanisms that can support bactericidal serum activity, and this has been nicely shown by Susan Roy-Jacker's group several years ago. And of course, what applies to Empaveli, the recently approved C3 therapeutic, applies to all the complement therapeutics that target C3. Everything has to be placed in the right context, and a risk mitigation strategy has to be followed, including vaccination against specific encapsulated bacteria, such as Neisseria, Streptococcus, and Haemophilus. And of course, antibiotic prophylaxis is also in the picture, depending on the clinical protocol and application. And I would like to finish and acknowledge this phenomenal team effort that has been placed across the world with more than 130 collaborators that have contributed to the molecular and structural characterization and preclinical evaluation of the compstatin-based C3 inhibitors. And of course, this would not have been possible without the inspirational example and the leading effort of Professor John Lambris, who has been the discoverer of compstatins, these C3-based therapeutics that were recently approved by FDA. Also the founder of Amindus Pharmaceuticals, which is developing third and fourth generation compounds for clinical applications, and the founder of Aegean Conferences. And I would like to leave you with this slide, inviting you to mark your calendars, save the date, and hopefully join us in the next Complement Therapeutics meeting, which is going to be held live, if everything goes okay, in Rhodes next summer in June. And we're really inviting you to attend this conference where cutting-edge research will be discussed in terms of, as it applies to the complement drug space, and probably the novel insights from the ongoing clinical efforts in evaluating complement therapeutics will also be discussed at this venue. So thank you so much for your attention. I would really be happy to take any questions, if any.